Hello everyone. So I try to make some videos as a support for our course, and pretty much the first time that I'm making videos, so I hope that I will more or less manage to do what I intend to do. Let me first share my screen. So I think this is the last slide that I presented to you about CRISPR technologies in my little PowerPoint about CRISPR technologies. And the point I wanted to make here is that actually, if we want to use these CRISPR technologies, in most cases, we will use plasmids. And in this case, we will have to manipulate these plasmids. And this means that we will have to work with quite complex plasmids with many different components encoded. And this is why I would absolutely think that it is required to use one of these softwares to properly be able to manipulate these. So we have SnapGene, we have Genius, and we have CLC Workbench, Genomics Workbench, or Main Workbench. And my favorite program is the CLC Workbench, but this is just because I'm using it for years. So every one of these softwares, I think, is very nicely adapted for editing and working with sequences. And Genius can also treat next generation sequencing data, just like the Genomics Workbench. And the main workbench that we have been using in the course and I want to present here now is just, I think, more or less the cheapest of all solutions. So if we want to use this, we have to get the software. So if I just switch to Chrome, so if I search for CLC Main Workbench, obviously I did that before. I come to Qiagen, I can click on download a free trial. And here we can also get, for example, courses provided by Qiagen directly. If you really want to download the software, you have here a button, request a trial. You need to fill this personal information and will receive a download link afterwards. So of course, did already install the software. I will just open it on my computer. We are now at version 24.0.1. And if you install the software on your machine, you will obviously have a database. So this is your database area. This is where your files are, which is in the first place empty. So I'm using it since years. So I have several different places where I store data. So also normally a network partition, which I use to share data with my collaborators. So CLC Workbench, as I said, we have the database here. So this is where all the files are, and we can have different types of files, so sequence files, protein files, PDB files, AV1 files, and so on that we import into the software. And the moment it is imported, it is converted into something that is in the file explorer abbreviated with .clc. So if I try to go to the place where we normally where we have this on the hard disk. Where is this? I think I can normally show this somewhere, but I don't find it. So just go there. So users and then CLC data. So if I go to users and then my identification, then I should have CLC data here. And you can see that all the files are .clc files. So this means this is a proprietary format, but we can always convert it back to whatever it was in the first place. Okay, so nothing is lost. So this is our database area. Up here we have a menu, we have a couple of buttons and a couple more buttons here. And we have down here an area that is dedicated to commands. So like aligning sequences, doing in silico cloning, making multiple sequence alignments, and so on and so on. So first of all, let's just speak quickly about the format of sequence files. So if I go to NCBI, I don't know why I do have this. So I just search for a gene and use GUI. from Pseudomonas aeruginosa as an example. If I search for this and open this file, what I see, I should not want this. So we come here. And what we see now is what they call here full report. And we have a number of different things. We can also from here come to the strain. So here we would have the full strain and the full genome somewhere. I actually don't know where exactly we have it right now. So normally, so this is now an entry for a DNA file. And we have down here now the different things. So the genomic sequence, the mRNA and protein. So if I go to the genomic sequence, open it here as GenBank. This is where I wanted to come in the beginning. So we'll have something looking like this. So this would be a GenBank format file. So I can say that I want to send this, the complete record, to a file in GenBank format. I say create file, and now it will download the file here. And if I look at this, so this is now something that has a .gb extension, and I can open this in, for example, in a Notepad or block notes. And you see, this is just a simple text file. And actually, the file will basically work once we have the header line with these couple of fields. And we also need the end, which is this double slash. And what comes in between can be quite variable. So it depends actually on the annotations that are made on the sequence. What are the annotations? So all these different lines are the annotations, and we can look at this in a graphical view. So where is this? Where is this? Graphics. So we would here now have a graphical interpretation of this GenBank file. So these are nice files. There are a lot of information in there. The thing is that, of course, as long as we are on NCBI, we cannot change anything of this. And as soon as we have opened this as a text file, it is also very bulky. So if I want to, for example, add something here, it doesn't make much sense. It won't help me. And this is where actually the softwares like CLC Workbench and so on come into play, because they help us to visualize these files and to also edit them. So how do I get a file into the software? Different versions, different possibilities. I will for today just make a new folder here, which I call demo temp, and I will delete it later on. So since I have downloaded the file, it should be in my downloads, and I can normally just drag and drop it. So this sometimes doesn't work, but normally it should work. And then I would now have this file here. What I can also do, so we have an integrated, we have of course an integrated search on NCBI. I can go on this download button, search for sequences at NCBI. And now, since I already had the very same one, I go back to the GenBank entry. I can use this identifier to get exactly the same sequence. Delete this. And normally I can then directly import it. Oops, that's not what I wanted to do. So down here you have a button that says download and open. So I can do that. And now it will open a second tab in the split view where we will now get this sequence downloaded. I think it's actually downloading the complete genome of Pseudomonas aeruginosa. So this is why it takes a little while. While the text file I had before was just a little entry within the genome. But let's just maybe wait for the genome to download. This is not so bad as an example. So here we are. So what is nice about the genome, I close this window about the NCBI search, is that we now have the full sequence of our organism with all the annotations. So all the genes are annotated, yeah? So the green arrows, the yellow-greenish arrows here would be coding sequences. So from the ATG start codon or whatever, these are bacteria. So we have several different start codons until the stop codon, where the blue arrows are gene annotations. So in a prokaryote, this is the same because we don't have introns and UTRs are not annotated. But in a eukaryote, normally this will be quite different. Yeah, so if I now want to save this genome, I can put it, just drag and drop the little tab from here into my folder that I just created. And now I will have it here. I can close it and reopen it. So you will realize that when I open the sequence file, I have a ribbon that opens here on the right-hand side. Just once again, now there's nothing. And as soon as I open the sequence file, I have this ribbon here. And actually it depends on the type of file, what options are visible here. So this is a DNA sequence file. So we will have a number of options, as for example, the wrapping of the sequence, so whether it's line breaking or not. Yeah, so I can say no wrap, and it will be just a single line. We can also have, since it's DNA, a single-stranded or double-stranded view, and some numbering options and so on. Then this is very important. So I said in the beginning, in the gene bank file, we have these annotations. So we have actually here gene coding sequence source. These three were in these features. These three were in the text file that I made, and these are now hidden here. So annotation layout just is the way they are shown on the screen, and annotation types shows us all the different types of features that are contained in the file that I just opened, and I can either tick the box to see them or tick them to hide them, untick them to hide them. Then we have here another entry about restriction sites, so I can decide to show or hide different restriction sites. And since this is now an entire genome, it doesn't make an awful lot of sense to show the restriction sites. But nonetheless, even with these couple of megabases, it's still possible to show them. If you have, for example, the Arabidopsis genome open in the software, it's not going to show the restriction sites anymore because the sequence is too large. Then below, there's a thing called motifs. I actually never use this. There's a thing about residue coloring. So if you prefer to have this very colorful, there you go. I think it just makes things more complicated. And then there is a very important tab here that is called nucleotide info. So once again, we have some things that are never used, like quality score, color space encoding. But this year, translation, this is actually very nice because we can show our protein annotations, our protein sequences, and we can now select whether we want to have a translation of any feature that is annotated as ORF or coding sequence. You can also say we want to see the reading frame +1, all forward reading frames, and so on and so on. So this is the default selection. And then there's a box find, so we can search for any type of sequence. So let's look for a sequence in this format, and we will find it, of course, in a genomic sequence somewhere. 10 base pair sequence should be present, and it's only present once. You can also search in the annotations. So in the beginning, I looked for the gene GUI, so we should find that name somewhere in the gene annotations. Here I have it. And we can search for a position. So if I would, for example, extract a position from somewhere, so you see that when I downloaded GUI from NCBI, I actually downloaded just a little fraction of the genome. The coordinates are given here. So if I take this starting coordinate, I should actually get this very end here because it was in this direction. So if I say position and I paste this number, position up, then I end up here at the very end of GUI because that was the left-hand coordinate. And down here we have a last thing that is called text format. So we can, depending on the screen resolution and what we want to see or not, can make this larger, make it smaller, and also change the font and so on. And indeed, these view settings, so I actually already adapted them, and it opens with what I saved as default view settings. So for every type of sequence, you can save one or several types of view settings and then apply one of these as default view settings. So how do you do that? So now I change the font size. I can say view settings, save view settings, and I call this now large font. And if I want to reuse this with other sequence files, then I say that I want to save this for use by sequences. And if I always want to use this, then I say use as default view setting for sequences. I actually don't want this because it's a bit large. Let's just save it. And if I open something different, you see it's now small. I can say that I now want a large font. And there I go. Now I have these view settings applied to the sequence file. And this is actually quite important because you will adapt the view settings to what you normally use. So I don't want to, among the annotation types, there are several that I don't consider tremendously important. So they are not shown by default. And also in the restriction sites that are shown once you have a file where it does make sense to show them, you can select which ones you want to have shown. So I just close this one again. I go back to this Pseudomonas aeruginosa genome. So this is, for example, a selection of restriction enzymes that we have in the freezer. So everything we have. And of these, I selected some that are by default ticked, so shown or not shown. Yeah, and you can save these settings. Thing is that you have to do this for any type of file. So if you, for example, you now have a circular view. So this is a linear view. If you have a circular DNA view, then it will be a new set of view settings actually. Okay. So if now I want to work on a specific gene, this is now all very large, but it doesn't matter. Oh yeah, maybe zooming, unzooming, and zooming. So you can either unzoom and zoom with this slider here. So whoop, whoop. Or you can press a plus and minus, of course, to do the very same thing. Or what is a lot easier, press Control on your keyboard and then just turn the mouse wheel and you will actually zoom and unzoom with the mouse wheel, which is very convenient if you want to, well, go into and out of your sequence regularly. Tack, tack, tack. And the same is true. So if I put the view settings to no wrap, now I will have a long horizontal sequence displayed. Now I can use Shift and the mouse wheel to actually go move in my sequence. Yeah, so I can zoom out. I can go to the left or to the right using my mouse wheel. So here we still have GUI. So I was just using this as an example, but if I go back here, and I now don't want the entire genome, maybe I want to have a bit of upstream and a bit of downstream sequence of GUI. I can just mark this. So with the left mouse button, I can select, and then I can make a right click on this sequence, and I will get a context menu. Again, just like with any type of software today, the different things shown in the context menu are, of course, sometimes different. So now I can say move starting point to the selection start. I don't want to do this. This would just shift the numbering of the sequence and so on and so on. And I can do edit the selection. So I could now really alter the nucleotide sequence. But what I want to do is actually to open it to have a small file, something that I can handle more easily than an entire genome. So what I do is to go here, open selection in a new view. Up. And now I get again a split view, just like before when I was searching on NCBI. And maybe now I move this here to make my life easier, to have it displayed more, bigger. Once again, so I'm a European and I'm a bit limited maybe sometimes with my brain. So reading sequences from the right to the left is really not an easy exercise for me. I want to have the ATG on the left-hand side and the stop codon on the right-hand side to not get confused. Also, it doesn't change anything. So if I want to do that, I can now make a reverse complement, of course, of this sequence. So either I make a right click I can go now on Toolbox, Nucleotide Analysis, Reverse Complement Sequence up, and it will ask me which will open this kind of guide. So which sequence I actually want to reverse complement this selection. It by default always takes the sequence that was just selected as a tab here. And then I say next, asks how to handle the results, so either to open it or to save it. So I just want to open it in a new file. So now finally I have, well, it's not an ATG, it's a GTG, but still the start codon on the left-hand side and the stop codon on the right-hand side, as I'm used to see this as a European. I have BPI1 sites displayed. Maybe I don't really want this, so I can turn them off here up. So if you would do the same thing, you would not have BPI1 sites displayed because you did not select these enzymes. If you want to change the enzyme selection, you can click here on Manage Enzymes, and let's say I eliminate BPI1 now. So on the right-hand side are the enzymes that are shown in the panel, and here is a list of all enzymes, which I can also modify, so I can make enzyme lists and change the content of this left-hand side. So now if I take out BPI1 from the enzyme shown in the side panel, then it is now gone. It should be here under multiple cutters. Now I can, of course, put it back in by a double click or by clicking this arrow. And now it will be back here with 3 cut sides. So this is basically for this right-hand ribbon of sequence display options, okay? Then we have all the tools, and I think actually the nucleotide analysis is some often very helpful. So reverse complementing, for example. And then what I use a lot are actually Sanger sequencing analysis if I want to compare sequencing files with a reference, and also this cloning tab. I don't say that the rest is useless, but I mainly use these 3 different, the commands that are contained in these 3 different menus. Okay, so now we still have a linear display. This is, of course, this is a genomic sequence, so it is linear, but I can just for fun show it circular. So then I get a little error message here. The sequence is not circular. Do you wish to make it circular? I can do that. Why not? So now I would see GUI and a little bit of flanking sequence in a circular view. Doesn't make an awful lot of sense, I agree. But still, for plasmids, it does make sense. And now it is safe to be circular, and some things will, well, show a weird behavior. So I can make a right click on the name of the sequence and select that it is linear once again. So now I don't have, you saw that something changed here. So actually the view settings for circular and linear views are different. But nonetheless, this is the circular view. So for working with plasmids, this will be helpful to have an overview of the different features of a plasmid. Then we have here a table of the annotations. So this is not super interesting right now, but it could be. And we also have this tab here where we go back to the initial gene bank view. What is interesting about that? So I said in the beginning, this is a text file, and it's not so easy to edit, or it doesn't make a lot of sense because it's not intuitive. Here I can now make new things, put new things on my sequence. So maybe this here is a super interesting GC stretch for me. So I can say, I can mark this. I make a right click on the selected sequence. And I can now go here and say add annotation. So I will call this GC stretch. And I will maybe make this a feature of the type motif. And I have here the orientation. So maybe this could be also on the other strand. Okay, so now I add this annotation, and if I go to this gene bank view, then I will of course have this new thing in my file normally. I didn't save. How do I save? I just save a copy once again. Oh. close everything else. And now normally here in the annotation types, I should have motif. And if I say that I want to export this as a GenBank file, maybe I want to send it to a colleague. And then I don't want to send the CLC file. I will send a GenBank file. I can press Control E, or I can go on export here. I select GenBank, and I use this one now. And I output this to a folder on my desktop. This is CLC export. And if I go on my desktop and open this folder, what was this thing called? NC0025. This one here. So if I now open this with block notes as I just did before, it should now contain this new annotation of the type motif. I hope so. Yes, yes. So it's a miscellaneous feature of the type motif, and it's called GC stretch. So this is now new on my sequence, yeah? And it will be shown by any of these sequence viewers if I want to. Obviously, this is not very helpful. I delete it for the moment. What is very nice is that when I edit the sequence, this is also maintained in a history. So maybe I induce a mutation by site-directed mutagenesis to eliminate one of these PPI1 sites. So maybe I'm really not very familiar with the rules of DNA, and I just randomly put a T here. So if I have only one single nucleotide selected, I can just press A, G, C, or T to modify the base at this very position. If I have several selected, I cannot delete and replace. So here I would have to say now, select these in the stretch, make a right click, and I say edit selection. And maybe I say N at this very position. So of course I can press Control Z to undo this, but maybe I just save this by mistake and so on and so on. And then things are weird. Things don't work in the lab anymore. And then I should normally be able to follow this up. So I have here the things that happened. So replaced single symbol. So I will see here that I replaced an A at position 251 with a T. And I also see that I have modified this sequence stretch to become this sequence stretch. So all of this is actually saved in the file history. And if I did not delete the genome in the beginning, then I also see where it comes from. So this selection, yeah, it actually originated from this whole genome assembly, which I downloaded in the first place, which in this case is still there. So I can click on it and it will open up. And this is actually very nice to keep track of what you did, and especially important if things are not as they are supposed to be. I press Control Z or undo. So it would be edit undo to come back to the three, eliminate these three ends. Let's just see what I did with this A to T exchange that I just randomly inserted. So I can turn on the translation to see the sequence of GAK, and I might actually have been lucky. So if I press Control Z again, it comes back to GAA, and this was coding for an E. And so I was not lucky, so I redo it. Redoing is Control Y. So I changed this to a D, to an aspartate. So this, I saw that I might have been lucky because it was the vowel position of this triplet, but in the end, it was still a non-synonymous exchange. So if I had done this to just eliminate the restriction site by site-directed mutagenesis, I would have done a very bad job, because I would have induced a mutation in my protein sequence. So I come back to the original sequence. So this is the yellow arrow, as I said, is a annotation of the type coding sequence. So if I mouse over it and wait a bit, we get this context menu displayed, and it actually shows all of the different entries in this translation. So what does that mean? So if I make a right click and I say edit annotation, I get such type of a window opening where I do now have all these different entries. So since this comes from a NCBI genome release, I have actually all the, basically the information where this comes from, and we also have a translation of this very gene saved within the annotation. So there is an interesting functionality. So at least there has been. If I press now, or if I make a right click on this and I say translate, then it proposes me here two different things: select the genetic code translation table or extract existing translation from annotation. So what does that mean? If I use this, it will translate reading frame 1, I think, in this case. No, actually, it will translate the gene annotation. While if I use the other option, it will use what is saved in the annotation history, in the annotation information. So this is important as soon as I start working with the gene and maybe making intentional mutations. Because then if I use this, it will still use the original translation, so the wild type sequence. While if I have changed amino acids, this will not be displayed in the translated protein sequence. So in this case, you need to, let's say I changed this here to an A. It's now a methionine. So if I say that I want to translate this using so this is approximately amino acid number, well, it's actually number 5. So here you see that number 5 still is leucine. While if I say that I want to translate this using this option, then I should not now have the methionine here. If I have, so this would be now a protein sequence, I can of course also save this in my database. So I just drag and drop the tab from here to there, up, and now the symbol is different because it's a protein. I have this also in my database. It's actually protein with a mutation, so this may not be the greatest thing to do. So I delete it, so just press supprimer, delete, or throw it in the recycle bin, or do whatever you want. Control Z to get rid of this mutation. And now I'm happy with this sequence. Okay, so I think I've gone through the different ways you can have your sequence displayed. And with that, I would like to come to a new point, which is basically in silico cloning. And I will do this in a second video, which I will start in just a second. Thanks.